touching.
Good morning. Today we are going to discuss an article with the title Glycosylation of Serine Prion-rich intrinsically disordered regions of membrane-associated proteins in Streptococcal. Since we have learnt about the intrinsically disordered proteins, now we are going to see an intrinsically disordered region of a membrane-associated protein in the Streptococcal. This journal was published in the this article is published in the journal Nature Communications with an impact factor of 15.7 in 2025. So what does this title mean? Glycosylation. Glycosylation is a biological process in which sugar molecules are enzymatically attached to proteins or lipids. This modification affects protein folding, stability, signaling and cell interactions and is essential for many cellular functions and immune responses. So what does the title say? Glycosylation of serine prion-rich intrinsically disordered regions of membrane-associated protein in Streptococci. So in the Streptococci bacteria, they have the membrane, and in the membrane there are some proteins, and these proteins are glycosylated in the serine and prionian region, where there is an intrinsically disordered region. So this is the title. And the novelty is protein disorder in prokaryotes has not been systematically identified and characterized even in model organisms such as E. coli and Bacillus subtilis. So the aim of the study is to identify the mechanism utilized by the S mutants to modulate biofilm synthesis and provide a roadmap of future functional studies of glycosylated IDRs in prokaryotes. So the main thing is how this S mutants modulate the biofilm through the IDPs in the IDRs in the proteins which are in the cell membrane. So in the materials and methods, we see the bacterial strains, growth conditions and media are all given. So these bacteria are used in the study, and they did construct the mutant streptococci also. So the in vitro analysis of synthetase activity of all the analysis were done. Then the co-sedimentation assay with fluorescent lectins were done. Fluorescent DIC and microscopy was done. Isolation and purification of membrane glycoproteins. So they need the glycoproteins on the membrane of that particular organism to study. They did analyze the proteins through LCMS, and immunoblot analysis of cell lysates were done. The biofilm assay was done since they wanted to find the modifications which is undergoing in the biofilm. And the thermal protein stability assay, circular dichoism was done, which is a spectroscopic technique. And prediction of extra cytoplasmic IDRs. Finally, the statistical analysis were done for all these techniques which was followed. The 2-day unit test and 1-way ANOVA with the QT's multiple comparison tests were performed. A p-value equal to or less than 0.05 was considered statistically significant. And in the results, they what they found, they identified identification and glycosylation of extra cytoplasmic IDRs. So when we talk about extra cytoplasmic, so this is the cell membrane. Outside the cell membrane, these are the extra cytoplasmic IDRs, which are C. So we see these regions are the C terminals. This is the N terminal. The C terminals have the IDR regions. These are the linkers and spaces. I talked about linkers and spaces. These are the linkers. So this is the membrane associated genome-wide analysis identified extra cytoplasmic serine threonine. Serine threonine are amino acids, which IDRs in several biologically important membrane associated proteins across various Streptococcal species, including S. pyogenes, pneumoniae and mutants. These IDRs are often located at the C termini of membrane associated proteins and are characterized by polyserine threonine-rich residue tracks. So this is the basic information about the membrane-associated protein and where the IDRs are located. The glycosylation S. pyogenes and S. pneumoniae, what happens? So the IDRs in S. pyogenes and pneumonia are glycosylated with glucose. This process is mediated by glycosyl transferase. So this is how we have to understand they are getting glucose is getting attached like this. So when we see this is the protein and the IDR, where the yellow ones are the glucose which are getting attached to that, which helps in the protein folding under stress and forms the biofilm. When there is no glucose getting attached, there is proteolysis, and this prevents the aggregation of the biofilm in that region. So this is what they wanted to do. And what is the functional importance of glycosylation? The O-linked glycosylation proteins, IDR containing proteins, form proteolytic degradation. And the absence of glycosylation leads to increased proteolytic processing of proteins like the PRSA and the PPMP, suggesting that glycan shield these regions from proteins. So the glucose molecules protect the proteins from being degraded. When they are not there, they are getting degraded. So role in biofilm formation. So glycosylation is critical for biological function of PRSA. PRSA is nothing but that protein. And the study found that the absence of glycosylation leads to a defect in biofilm formation under ethanol stress conditions. PRSA is a membrane-anchored lipoprotein chaperonide which helps in the folding, found in the gram-positive bacteria. And the protein levels and stability was also tested. So glycosylation positively impacts the total protein levels of the several membrane-associated proteins. And the IDRs also positively contribute to optimal protein levels of all the organisms. Optimal length of PRSA IDR is important for its efficient glycosylation and maintaining protein levels. So these were the results. What were the limitations? There were unidentified proteases for PRSA cleavage. How did the protein degrade? That was not mentioned here. And the mechanism of glycosylation's impact on protein function was not discussed. And scope of IDR function beyond the studied proteins. The paper itself states that it is currently unknown how the identified IDRs contribute to the activity of other proteins. The future scope is the paper explicitly mentions that future investigation of IDR functions may provide insights into how these regions sense and respond to environmental changes to boost bacterial survival. So concluding this article, this paper concludes that extracytoplasmic IDRs are a widespread and functionally important feature in Streptococci, and their own glycosylation is a sophisticated mechanism that protects these regions from proteolysis, maintains protein levels, and is critical for bacterial processes like biofilm formation, especially under stress conditions. So the article, although the study did not follow any standard reporting guidelines like STOVE, consort, or ARI due to the experimental nature, its methodology aligns with the field appropriate best practices in molecular biology and enzyme biology, which follows this trend that is standards for reporting enzyme data, which is being followed in these designs, which includes the fundamental principles of experimental design, including use of appropriate controls, replication, and validation techniques. Thank you.